Hello, my name is Alex Cabot, and this is my science fair. How quickly does the cleaner, force in nature, lose effectiveness in killing common bacteria? The question I was trying to answer in this experiment was, how quickly does the natural cleaner, nature's force, lose effectiveness in killing common bacteria? The fascinating fact about bacteria is that bacteria can survive on radioactive waste. If radioactive waste does not kill bacteria, then how do we kill the bacteria? The question in this experiment that I was trying to answer was, how quickly does the natural cleaner, nature's force, lose effectiveness in killing common germs? The hypothesis for this experiment was that if the natural cleaner, nature's force, is used over several days, then it will be most effective the day it is first activated. The cleaner was activated right before the first testing. 3 different petri dishes were swabbed at the same time using 3 different desks. There are 3 different areas on each petri dish. This experiment was done 2 more times. The second testing was done the same way, except the cleaner was activated 24 hours before the testing. The last testing was again done the same way, except the cleaner was activated 48 hours before the testing. Finally, all petri dishes were incubated for 24 hours immediately after the testing, and results were taken by scanning with the app called Colony Counter. The hypothesis was correct, because the bacteria grew when the cleaner was older. What could have been done to further this experiment was to use different cleaning supplies to see which is most effective. The hypothesis for our experiment is if the natural cleaner, nature's force, is used over several days, they'll be most effective the day it's first activated. My independent variable is the amount of time the cleaning supplies was used after it was activated. The dependent variable is the effectiveness of the cleaning supplies changed. The control was the area on the petri dish that was left untouched. And the constants were the same type of petri dish, the same type of agar, and the same temperature in the incubator. There are 4 different types of bacteria: spiral, concoli, rod, bacilli, spiral, spiroli, coma, burros, and corkscrew, spirochetes. I learned that on average there are 400 times more germs on a desk than on a toilet seat. The cleaner that was used in this experiment is supposed to be most effective when it's used right away. The active ingredient in the natural cleaner is hypochloric acid. The active ingredient in the natural cleaner, force in nature, is the same active ingredient commonly used in wound, eye, and veterinary care products. To activate the cleaner, you must first fill the Force of Nature's activator with tap water. Then add 1 Force of Nature's tablet capsule, push the button, wait about 6 minutes, then export to a spray bottle and use within 2 weeks. However, bacteria are called pathology bacteria because they cause disease and illnesses like strep throat, staph infections, tuberculosis, and food poisoning. The bacteria grows at 36 degrees Celsius. Some of the most common bacteria is Escherichia coli, Calbacilla phomia, and Staphylococcus aureus. Some helpful things that bacteria can do is that bacteria can help to digest food, destroy disease-causing cells, and give the body much-needed vitamins. Bacteria are also used to make healthy foods like cheese and yogurt. The type of agar that was on my petri dish was nutrition agar. My materials were 9 petri dishes, 18 sterile swabs, 3 desks, 1 bottle of the cleaning supplies Force in Nature, an incubator, a Sharpie, a phone, and the app called Colony Counter. This is my procedure. Take 3 petri dishes, and with a Sharpie marker, divide them all into 3 different areas on the petri dish. Area 1 will be the unclean sample. Area 2 is the clean sample, and Area 3 is the control. Take a swab and dip it in the bottle of water. Take the swab and take a sample of the bacteria on the desk. Put the sample on one area of the petri dish in a zigzag pattern. Activate the cleaning supplies, Nature's Force. Clean the desk with the Nature's Force by repeating steps 4 and 5. Repeat steps 3 through 5 with the desk you just cleaned. Repeat steps 3 through 8 2 more times with 2 more different petri dishes. Repeat steps 1 through 9 24 hours after the first test was conducted. Use the same Nature's Force that was used in test 1 and do not reactivate it. Repeat steps 1 through 9 48 hours after the first test was conducted. Use the same Nature's Force that was used in test 1 and do not reactivate it. Observe and collect data using the app called Colony Counter. As you can see right here, this was the first test. There wasn't many when it was activated, but here it was not cleaned, and there was a lot of bacteria that grew. Here, again, there's more bacteria than the cleaned sample right here. And again, overall, there's more bacteria here than in the clean sample. But you can see that throughout the entire process, more bacteria grew when it was older. This is one of my graphs compared to the data tables up above. This is the second one for test 2, and this is the third one for test 3. As seen in the graphs above, it was analyzed that the force in nature that was used right away was always most effective in preventing the bacteria from growing. The results were scanned by the app called Colony Counter and put into the graphs above. The question in this experiment that I was trying to answer was, how quickly does the cleaner, force in nature, lose effectiveness in killing common bacteria? My hypothesis in the experiment was that the cleaner, force in nature, would be most effective when it is used right away. In my experiment, I originally wanted to test the cleaners a week period apart, but ended up not having the time. So I did the experiment 24 hours apart. To start the testing, I swabbed 3 desks, 1 desk per petri dish. I divided each petri dish in 3 different areas. One area was swabbed from the unclean part of the desk. The second area was swabbed from the clean part of the desk, and the third and final area of the dish was the control. I put nothing in this area. For the testing, before I cleaned the desk, I took a sample from that desk and put it on the unclean area on the petri dish. Then I cleaned the desk with the cleaner, force in nature, that was just activated, and took a sample from the desk I just cleaned and put it on the cleaned area on the same petri dish. I incubated and then repeated these steps to plate 2 more petri dishes. After I incubated the petri dishes in an incubator at the temperature of 36 degrees Celsius for 24 hours, then I took the results. 24 hours later, I did the same thing but did not reactivate the cleaner. Then, for my third and final test, I did the same thing and used the same cleaner, but didn't reactivate it. My hypothesis was correct, because the cleaner that was older was most effective in preventing colonies from growing. If I were to do this experiment again, I would future them, I would use different cleaning supplies to see which one is most effective in killing germs. This is one of my works cited. This is another, and this is my last one. Thank you for watching. My name is Alex Gabo. Thank you.